So our next talk is going to be by Elisabetta Soragni, and Liz is FARA's new Associate Director of Research. So she came on full-time in January, but Liz has been working in the FA space for about 10 years, and just making the transition from the lab bench to FARA to help us advance a bunch of our research initiatives. And so I'm really excited to have Liz here today, and she's going to take you through a little bit deeper dive into the molecular mechanisms of FA and the various treatment approaches that go with what we understand about FA.
Okay, so good morning, everybody, and it's nice to meet you all. It is actually my second time at the UCLA Symposium. So we've heard from Dr. Perlman all the clinical features of FA. What I would like to talk to you now is more, as Jen mentioned, a little bit deeper into the molecular mechanisms and how this knowledge can inform us on possible therapeutic strategies. So you've heard from Dr. Perlman that FA was first described many years ago in 1863 by Dr. Friedreich, and about 13 years later he articulated the hereditary nature of the disorder. He thought that disorder was genetic, that it had a genetic basis. But it took about another 120 years for the genetic locus to be identified. And it was identified by Dr. Pandolfo and his collaborators. And they found that FA was associated with this location on the DNA that had this particular feature. It had a GAA triplet that was polymorphic. What does that mean? It means that it changed among people. So what is this GAA triplet? You all know that inside your cells, there's this very important molecule inside the nuclei of your cells. There's this very important molecule that is your DNA that determines a lot of who you are. It determines how tall you are, it determines what your eye color is, and this DNA, that is depicted right here, is made by only 4 very simple building blocks. And these building blocks are named by their initials. They're named A, C, G, and T. So what is the GAA repeat? What are the GAA repeats? Are just GAA, these three building blocks repeated over and over again. And what Dr. Pandolfo found was that in unaffected individuals, these repeats are very low. They're between 8 and about 30. But in people with FA, these repeats are over 100. And in most patients, they are between 600 and 900. So he also found that when these repeats are high, over 100, the locus that contains the repeat doesn't function properly. This locus encodes for a protein that's called Frataxin. And when the repeats are present, both the Frataxin mRNA and the protein are expressed at very low levels. So since this discovery, scientists have been trying to figure out why, when you have such a large number of repeats, the gene doesn't work properly. So to explain that and to explain what we know about this process, I'd like to make a little bit of an analogy here. So as I mentioned, inside the nuclei of your cells, you have your genetic material that determines all your genetic traits. This is your DNA. So how does your DNA determines how tall you are and what your eye color is? It does that by being copied into this other molecule that's called the messenger RNA, which is then transported from the nucleus to the cytoplasm, and it's used as a template to make proteins. And the proteins are what constitute a lot of your body. They are a structural component of your body. They participate in all the enzymatic reactions of your body inside the cells. So I like you to think of genes as roads, and of this process of making the messenger RNA or mRNA called transcription as trucks that carry their template or the mold on this road. This template is then, as I said, this mRNA transported into a different location in the cell, and then is used in this assembly line to make the final product, which is the protein. So what happens when the GAA repeats are, the number of the GAA repeat is not long, it's short. So the trucks can easily travel through the road, along the road, and they deliver their cargo, their template or their mold, to the cell assembly line. A lot of the molds are delivered to the assembly lines, and the assembly line uses the mold to make the final product, the protein. So a lot of the protein is made. So what happens when the GAA repeats are longer, over 100 or 900 or even higher? Well, we know that two things happen. The first thing is that the GAA repeat forms some kind of a roadblock. So our trucks cannot travel along the road anymore. And the second thing that happens is that these signals, the do not enter signs, are posted at the beginning of the road, so no more trucks cannot even enter the road. So when this happens, only very few trucks can get to the end of the road. They deliver very few molds, so the cellular machinery, the assembly line, can only make very little frataxin. So what are these roadblocks and what are these signs? Well, the roadblocks are called R loops. So as I mentioned before, your DNA is copied into this molecule, the messenger RNA, by a process called transcription and by an enzyme that's called RNA polymerase II. This enzyme, as it travels along the RNA, it makes the copy, the messenger RNA, that is our template, our mold. But when it copies the region that contains the GAA repeats, and when these repeats are high, these GAA repeats kind of get tangled with the DNA, so the RNA polymerase, our truck, gets stuck there. And so this constitutes the roadblock that prevents other trucks from traveling along the road. So what are the do not enter signs? The do not enter signs are signs of formation of what we call heterochromatin. So to explain this, you have to think of the DNA as being inside the nucleus, not as a linear molecule, but has wrapped around these other proteins that are called histones. And this is what makes chromatin, your chromosomes, a combination of DNA and this protein called histones. So when the DNA looks like this, looks like a pearl on a string, these little signs are posted on this histone protein, and these little signs mean the gene that is here is open and can be transcribed, and that mold can be made. But when certain enzymes are working on these molecules, they cancel, they remove these signs here that make the gene open, and they substitute these signs with this do not enter sign. So what happens is this structure becomes very compacted. So the genes that are in here are not open anymore, so they're not transcribed anymore. So the genes are silenced. And this is what happens when the repeats get long. All these do not enter signs get deposited on top of this histone protein, and the drug cannot enter the road, and transcription cannot occur. So how do these two mechanisms work together? We'd really like to know, because it's important to know, once we know the exact mechanism, how to target the mechanism, but we don't know for sure, but we can hypothesize that if we go back to our analogy, that when this roadblock appears, when the R loops appear, when you have long repeats, and when the first few trucks cannot travel along the road because of the roadblocks, a signal is transmitted at the beginning of the road, and these do not enter signs are posted so that other truck know that there's a roadblock ahead and the road cannot be traveled. So as I said before, very few trucks can get to the end of the road, few modes are made, and little frataxin is made. So now that we know all this information, how can we use this information to design therapeutic strategies that can restore the functionality of this gene? So one simple approach is, well, if we go back to our analogy, our trucks cannot travel this road because there are all these blocks. So why don't we provide a detour for this truck so that they can travel through a different route and reach the same destination? And this is pretty much what gene therapy is. And I'm not going to talk about much of it. Susan is going to talk this afternoon about it. But gene therapy is delivering to a cell that doesn't have a functioning gene, a functioning frataxin gene, so that this truck can travel through the road, deliver their cargo, and frataxin protein can be made. A similar therapeutic approach is to use stem cells. So there's a lot of hype and a lot of hope in this stem cell therapy field, but the field is also extremely challenging. There's a lot of unknown questions. So this therapeutic strategy is not simply delivering stem cells to a patient and then hoping that these stem cells go to the right places and become the right cells and cure the disease. The challenges in this field are many. So what FARA is focusing on is a very specific application of stem cells, and this is an example. In this specific application, you can take bone marrow stem cells and isolate a very specific kind of bone marrow stem cells, and then from a patient, and then culture them in vitro, genetically modify the stem cells to remove the repeats, reintroduce them back in the patient, and from what we know from mouse studies, this particular kind of stem cell become a specific type, differentiate in a specific type of cell that can then go to the organ that we want it to go, for example, the DRG, and support the survival of cells that are dying because they don't have frataxin. And from mouse studies, we know that one possible mechanism that these stem cells do that is by delivering part of their content to the sick cell. And they do that, and this is an hypothesis, by making these tubes that are called nanotubes and they use this tube to deliver their content. And what they deliver is mitochondria. We think that what they deliver is our mitochondria, and I'm going to talk about mitochondria a little bit later, but also Dr. Perman told you how important these organelles are to make energy inside the cell. Another strategy is, and we had a question earlier about that, is to use CRISPR-Cas9. So we heard earlier that the problem with the gene is the presence of this repeated sequence, GAA. So it's easy to think that if we remove the GAA repeats, then the gene becomes active again. And so we relieve the transcription of repression, we relieve the silencing. And to do that, there's this new tool called CRISPR-Cas9, which it's really like a scissor. It's really like scissors that can be directed to the GAA repeats and cut out the GAA repeat from the DNA. And we've shown, it has been shown in vitro that if you do that, then the gene becomes active again. It makes more RNA and it makes more Frataxin protein. So I talked to you about the roadblocks. So are there therapeutic strategies that we can use to target these roadblocks? The first roadblock is the R groups. And in fact there are, and this is what some of the oligonucleotide-based strategies are used for. These oligonucleotides are simply made of those 4 building blocks that made the DNA. They're just short, and they target the GAA repeats directly. So what happens is that the oligonucleotides bind the tangle, the R loop, the tangle between the mRNA and the RNA, and they disrupt that tangle, and the polymerase now can freely walk through the DNA, and the road becomes open again. The other block in the road are these do not enter signs that I mentioned earlier. So wouldn't it be nice if we could remove the do not enter signs from the DNA, from the histones that are bound by the DNA, and in fact you can. There are enzymes inside the cells that are responsible for putting the sign on the histones and taking them off. So we can design more small molecules that target this. They're called epigenetic writers and erasers, and reopen the frataxin gene. So reopen the road. Another way to restore the frataxin gene function is to use what are called synthetic transcription factors. And we're going to hear about, so I'm not going to talk about it much because we're going to hear about it in a little bit by Dr. Jeffries. And these molecules are molecules made of 2 parts. One part that binds the GAA repeat, and another part, the second part, right here, is a part that is this synthetic transcription factor. So if we go back to our analogy, we have our road that is blocked by the roadblock and by the do not enter sign. We have our little truck that cannot travel through the road because of the roadblocks. And what these synthetic transcription factors do is they make the truck a supercharged truck. So now the truck can easily travel through the road, get to the end of the road, and deliver its cargo. So let's switch gear a little bit. We talked about what the gene does and what the problems with the genes are, now let's talk about what the frataxin protein does. Okay, so the frataxin protein is delivered to this organelle, the mitochondrion, which, as Dr. Perman said, is very important in the cell metabolism because it's kind of the power plant of the cell. All the energy is made there. But what does the frataxin do inside the mitochondria? The frataxin itself is part of an assembly line. It has its own assembly line to make this other protein called iron sulfur cluster protein. And you heard that from Dr. Perman's too. So the frataxin works together with this other protein to deliver iron and sulfur to this iron sulfur cluster protein. So what are these iron sulfur cluster protein? Well, this is the cartoon of a cell, and here, highlighted in red and in green, are all different iron sulfur cluster protein in the cell. You see how many of iron sulfur cluster protein are in the cells, and how many aspects of the cellular metabolism all these iron sulfur cluster protein are involved with. So you can imagine how and why, once you lose frataxin protein, just one protein, you have so many different consequences inside the cells, and why you have so many different tissues that are affected in FA. So these are the consequences of the frataxin loss, and these are proteins that are involved inside the mitochondrion in energy production. So this iron sulfur cluster protein are not made because frataxin is lost, so you don't make energy anymore. So that's a big problem for the cell. Also, a consequence of the loss of this iron sulfur cluster protein and of low energy production is the elevation of this reactive oxygen species, which are inducing oxidative stress inside the cell. Also, because frataxin levels are low, the level of iron starts to go up, and this induces a process that's called ferroptosis, that is a cell death that is a consequence, a direct consequence of increased iron inside the mitochondria. Also, what happens when frataxin levels are low is that mitochondrial biogenesis is affected. What is mitochondrial biogenesis? Is making new mitochondria. So when the cell needs more energy, it makes new mitochondria because the mitochondria are the power plant of the cells, and without frataxin, the cell cannot do that anymore. So now that we know a little bit more about what frataxin does, where it is, what its function is, can we use this knowledge to think of other therapeutic interventions? So similar to what we said about the gene and how the gene is not functioning, so through gene therapy we're trying to give the cell a functioning gene, we can do the same for the frataxin protein. Except, so we can think of delivering the frataxin protein to the cell that doesn't have enough frataxin protein. This is not as easy, or it's not easy, because the proteins are big molecules, so they cannot really by themselves enter the cell. So that's why I have a little cross here. But luckily, frataxin, first of all, is a small protein, plus you can add this little tag to the protein called tag that helps the delivery through the cell, and with that little tag, frataxin protein can actually enter the cell. And this approach is called protein replacement therapy. So another way to approach the loss of frataxin is to think about not increasing the amount of frataxin inside the cell, not through making more of the frataxin, but to somehow reduce the degradation of the frataxin protein. So all the proteins inside the cell, when they are made, they perform their function, and then they are degraded, they are destroyed, and their components are recycled. And that happens with frataxin too. So there are this enzyme called ubiquitin ligases that deliver this little balloon, this little tag to the frataxin protein. And this little tag says, okay, this protein has done its function, now you can destroy it. And then the frataxin goes through this process and all the components, all of the frataxin is degraded and the components are recycled. So what can we do? We can find small molecules that inhibit this enzyme that transfer this tag, so that tag is not transferred anymore, so frataxin is not degraded. So if frataxin is not degraded as efficiently, its level in the cell remain constant, and it helps with the function, with its function. Another way of thinking of therapies that can help with the disease is trying to figure out a way of trying to target this assembly line. So I told you that frataxin is part of this assembly line in the cell that makes iron sulfur cluster proteins. So when frataxin is low, this assembly line doesn't work and you don't make the proteins anymore. But can we think of a way modifying this assembly line somehow so that this assembly line doesn't require frataxin anymore and the iron sulfur cluster protein are made? This therapeutic strategy is called identification of frataxin mimetics. So identification of molecules that can bypass the frataxin function in this assembly line. And this is actually a relatively new therapeutic approach, and FARA has been focusing on this type of studies in the last few years, and we have funded a few grants that are just trying to study just this, trying to find a way to make frataxin dispensable for the cell. And then finally, I explained to you how the frataxin loss has many different consequences in the cell. It affects so many aspects of the cell metabolism. And each of these aspects are an avenue of therapeutic intervention. For example, the ferroptosis, the cell death induced by iron accumulation, well, you can find molecules that inhibit that. And these are ferroptosis inhibitors. This is a brand new field. And also some things that you've heard, I'm sure you've heard, because these are some of the oldest approaches, like antioxidant or molecules that target the FIMPAR gamma pathway or NRF2 activators. OMAP is targeting the NRF2 pathway. So all this consequence of frataxin, and most of this therapy targeting consequences of frataxin are some of the most advanced therapeutic for FA. And why are they some of the most advanced? Because they're some of the oldest. And they are the oldest therapeutic approaches because all these pathways are also common to other diseases. So what FARA did at the very beginning is trying to learn from other diseases that involve mitochondrial dysfunction to try to transition that knowledge and apply that knowledge to FA. And finally, I'm going to leave you with this summary of what we talked about in the last half hour. We talked about going from identifying the GAA mutation, how the gene is silenced, what the protein does, and what the consequence of what frataxin does, and all the different ways that you can target all these different steps. And these are all very important therapeutic approaches. These are all worth pursuing, and FARA is pursuing all of them. The most important part is, and why we are pursuing all of that, because they're all important. but most importantly because we think that an effective treatment is going to be a cocktail of therapeutic, some kind of a combination of some of these different approaches. And with that, I'll finish, and I'll thank you for your attention.
Liz, thank you. Do we have questions for Liz?
Everyone.
Yep.
So from the point that the nucleus decides, hey, we need to make frataxin, from the point that the nucleus decides, or whatever triggers that the cell needs frataxin, to when the frataxin is made, how long does that metabolic process take?
So the transcription is only a few minutes. The translation can be a little longer, but within minutes you can make from when the DNA decides, or whatever is on the DNA decides. Okay, let's open up the gene. From that point to the production of the protein, it can be minutes to half an hour or something like that. So
Okay.
it's very fast.
So I'm assuming also then the cell has a way to regulate, hey, we've got enough
Yes,
protein
every
out there,
single
so
step.
stop.
Yes, every
Okay.
single step from the DNA all the way to the protein, the DNA to mRNA, so transcription and translation is very finely tuned and regulated.
Thank you.
So the oligonucleotide approach that you talked about,
Yeah.
I know there are oligonucleotide therapies for other diseases. How soon can we expect that to be in a clinical trial for FA?
Yeah, that's, I don't know. It's a great question, I don't know. This is still in preclinical development, so it means that it's still, these are still being tested in cell cultures and in animals, so the clinical trials are a little bit far there. Yeah, I don't know if you want to add something.
I can't give you a date, but I think some of the challenges still with oligonucleotides are having the cell take up the DNA. So when you deliver it, cells don't normally like to just take stuff up. So part of the challenge has been to see if there are ways to get them delivered more efficiently. And then the other thing is oligonucleotides, some of the ones that have been approved or are in clinical trials are blocking genes from being read. They're blocking translation because those genes normally make abnormal proteins. And it turns out that using oligonucleotides as activators is a little bit harder. And so the potency of the oligonucleotides as activators is a little bit lower. And so we've been trying to test more and more oligonucleotides with slightly different chemistries to increase their potency as activators. And so those are still some of those preclinical challenges that are being worked out.
There was an HBO show a year and a half ago where one of the East Coast surgeons really did it on purpose or not. He attached whatever it was he wanted to transfer into wherever he wanted to transfer into his tissue or heart or blood or bones, like I don't remember. But he used HIV virus as the vehicle to cross the membranes into the central nervous system.
That's what the protein replacement therapy is. That tag is a protein that comes from the HIV virus. That's what it's used for. So that helps the delivery, that helps the Frataxin protein go inside the cell. Yeah, that's exactly what it is.
I'll just stand up because I talk loud anyway.
Yeah, Liz can repeat your question.
Oh, yeah. So given that the how, long, and when is always sort of foremost on a lot of our minds, how is technology being used to speed up the research process?
The question is, how is technology used to speed up the research process? By technology, you mean discoveries?
Or whatever, computer power processing, I mean,
Oh, yeah. Well, for example, there's a lot of hype right now on artificial intelligence, right? And there are applications of artificial intelligence to science. I went to this seminar, and this investigator was talking about cages for mice. You know that we use mice to study the disease, right? That are disease models. And the FA disease models that are in mouse. And this investigator used artificial intelligence to monitor the mouse 24/7, and then he used an algorithm to try to define, let's see, what the pattern of behavior of the mouse are when the mouse is healthy versus when the mouse is sick, and use this algorithm and all these recordings of the mouse, of one sick mouse versus one healthy mouse, to try to find patterns that indicate, okay, this is a pattern that's specific to the disease. So now what happens when I put a drug in the mouse, when I treat the mouse with a drug, do this pattern change towards the normalization? So that's one way where technology and computer power is used to improve science.
thank you.
Yeah. Liz, we met with a company, right, in November that was.
Yes. Yeah, that's right. There's a company that uses artificial intelligence in that way. So they have this special algorithm, I don't know what it is, but where you can add to this algorithm all the data that has been accumulated, for example, in FA. In FA, there's been a lot of studies, they're called transcriptional profiling, where they looked at all the gene expression in different cell types in FA, comparing to the gene expression in cell types that are unaffected, and looked at the difference. What are the genes that are expressed in FA and the genes that are silenced or changed in healthy cells. And they use all this data that has been accumulated over the years, both in human cells, in mice, and they feed all this data to this algorithm, and they're trying to figure out what are the drugs that could be useful to treat the disease, or to target specific pathway in the disease that are dysregulated.
The other thing that I thought of when you asked that question is just the rate at which information is being shared. When we both started in this field, like when you wanted to get access to new scientific information, it came out in a paper journal. And now those paper journals still exist, but they're all now obviously electronic forms, and just even the time it takes to get a paper published has just been dramatically reduced because of the technology, and the time it takes to access that information within the scientific community is so much faster. And I think that's a huge advantage that we don't think of very often, but it is a real one from my perspective.
So theoretically, if replacement therapy was an option, it kind of begs the question, how often do you replace something?
Yeah, so for gene therapy, it's going to be pretty much a one-time shot. So for gene therapy. Protein replacement, well, there's still some studies that have to be done that I have to figure out whether the protein replacement triggers some kind of immunity, some kind of immune response. And we still don't know that. But in theory, the protein replacement is a treatment that you can continue to administer. for gene therapy, they're more, the immune response is more of a concern. So it's likely that once you administer it once, then
Given the earlier question about timing, how things happen, does that mean the placement would have to be happening over a very small frame? Or do we know enough?
well, we know that, for example, for the protein is made pretty fast, but it's degraded very slowly. So, like, it takes several days for the protein to be degraded. So, you know, if you administer the drug, you know, at some point, then you can wait a few days because the protein sticks around for a little while. So you don't have to make it. You don't have to administer it every half hour or something like that. So because it's pretty stable protein, so it takes a few days for it to be degraded and recycled.
Are there well-known ways to kill off a portion of the mitochondria in a cell? And if you did that, would the surviving mitochondria have more fission or make more mitochondria? Because we know that, you know, short-lived cells that split often are okay.
Yeah, so the question is, if you want to kill that portion of the mitochondria that is not functioning well, then make more mitochondria. Okay, so this is actually a physiological response. So when the mitochondria are in trouble, there's a process that's called mitophagy. So the cell normally gets rid of this unhealthy mitochondria and kind of recycle them and makes new ones. So there are therapies that you can think of trying to target that process to reduce the mitophagy and improve the biogenesis, the making of the new mitochondria. And the NRF2 pathway, targeting the NRF2 pathway is sort of a way to do that. So what OMAB and dimethyl fumarate do, what these drugs do, is that they increase the mitochondrial biogenesis. They're trying to make new mitochondria.
All right. Thank you all for your questions. Liz, thank you very much.